Hi, my name is Scamfer, and I've been doing research in the Murtaugh Lab. Our lab is interested in the development of the pancreas and also in diseases of the pancreas, such as pancreatic cancer. Pancreatic cancer arises from acinar cells, which make up the majority of the cells in the pancreas and which produce digestive enzymes. PTF1A is an important transcription factor in acinar cells. Previous research in our lab has shown that PTF1A is lost during cancer development, that knocking out PTF1A in mice results in faster progression of cancer, and that expression of PTF1A in precancerous cells actually can reprogram these cells back into normal acinar cells. So due to these findings, we wondered if PTF1A could act as a master regulator of acinar cell identity. We tested this by infecting fibroblast cells with a virus that can conditionally express PTF1A in the presence of doxycycline. We hypothesized that PTF1A alone would be able to promote expression of target genes, but here we found that PTF1A could only promote expression of one, CPA1. There are a couple hypotheses that can explain these results. One is that PTF1A is unable to access the promoters of its target genes due to their location in heterochromatin of fibroblast cells. But another hypothesis is that PTF1A needs other acinar transcription factors to bind its target genes and to promote expression. We already know that in acinar cells, PTF1A works with a binding partner called RBPJL, and there's also a lot of other important acinar transcription factors that could aid PTF1A in promoting acinar identity. For these reasons, we hypothesize that PTF1A needs other acinar-specific transcription factors in order to reprogram for acinar cells. To test this hypothesis, we used mouse fibroblast lines that were already stably infected with conditionally expressing PTF1A or EGFP viruses. We then infected these cell lines with viruses that would conditionally express single genes, which are listed in the figure above, and also with two combinations of these genes called RMN and RMNFP. All of these genes encode for acinar-specific transcription factors, with the exception of an M-cherry infection, which was a control. Once stable lines were established, we induced gene expression by treating cells with doxycycline for 48 hours. Cells were then harvested for RNA, which was reverse transcribed into cDNA and used for qPCR to determine gene expression levels. So looking here at our first gene expression graph, we're looking at expression of acinar-specific transcription factors to see if our viral infections worked. Wild-type acinar levels of expression are set at 0, and so we can see that our PTF1A plus dox samples are all being expressed at or above wild-type level. We can also see that RBPJL is being correctly expressed in all the lines that it was infected into, and we see that NR5A2 is being correctly expressed in all lines except GFP-RMN. So it looks like NR5A2 may not have been incorporated into those cells, but luckily our GFP lines are just a control. Our second gene expression graph is looking at expression of PTF1A target genes, which are acinar digestive enzymes. On the whole, it looks like different combinations of transcription factors are able to better promote different acinar target genes. For example, our PTF1A RMNFP combination is able to promote the highest expression of CPA1 and possibly CELA2A, while it cannot promote expression of amylase 2 here. However, our PTF1A RMN combination is able to promote higher expression of amylase 2, and it's possibly also promoting expression of elastase down here. We can also see from these graphs how individual transcription factors are affecting expression. For example, in CELA2A, we see that transcription factors contributing to higher expression are probably FOXA2, NR5A2, and RBPJL. So far, our data supports our hypothesis that different combinations of acinar-specific transcription factors with PTF1A will promote expression of different target genes. Future directions for this experiment include repeating the experiment for validity, trying different combinations of transcription factors, applying chromatin-altering drugs to cells, and expressing transcription factors in a specific order to emulate the in vivo process of acinar development. This figure here shows the Acinar differentiation process and what transcription factors are highly expressed at which stages. So we can see that PDX1 and FOXA2 are actually expressed earlier in this process. And And we think that imitating this natural expression order might give us higher expression of more of our Acinar target genes. Better understanding Acinar differentiation and what transcription factors play a role in this process will help us to understand both what's changing when Acinar cells become cancerous and how we might be able to treat pancreatic cancer by a redifferentiation technique. A big thank you to the Murtaugh Lab, especially Diane and Charlie, for their mentorship on this project, and thank you for watching.